rain samples and air samples in the Delta, we can most certainly suspect that if we're bordering the Delta, that some of those contaminants will disperse into our air as well. Some other things.
The fact that they had to go to a court case specifically for them, or maybe they've been sent to a juvenile detention center or a community service project because of misconduct. Any youth.
So when you have high blood pressure as a parent, it goes to your baby, brings all the glucose, it brings the stress on the baby. You have the heart doesn't form correctly.
Pretty much, I analyzed all of the 5 peer-reviewed studies and in my qualitative review, and I just kind of grouped them off of the
doctor in a sterile environment, such as a surgeon setting. So I feel like if we figure out what these microbes are doing gene-wise, we can potentially. In
recent years, there have been a rise in police misconduct in various states in the United States. Many people believe in the community,
which basically means the practitioner use these questions in a more broader term. That way, it's the patient's responsibility to interpret it and guide it in their way. Closed-ended questions
are already in use, and they've been put to use in crops such as rice, maize, and to that end, we need more sustainable agriculture.
Suicide ideation. Suicide ideation is the deep consideration or having a plan.
One of the leading diseases of concussions is CTE. CTE is a progressive brain disease that happens after repeated traumas to the head. And
thought on AI and just analyze not really the history, but just like how it has helped you in your academic career or even just your personal life.
The expectation factor, so anything that was being asked of them throughout like their process. And then in yellow,
another thing that many people think are the same: normal grief versus complicated grief. Normal grief can last up to 6 months and is typically more mild. And symptoms can include you're able to understand that you're
So when putting the context and looking at the Lafayette Oxford community, I want to think about 3 different things that could also contribute to pollution in this area specifically. One of the facts is that we're neighbors right to the Mississippi Delta. And if you know anything about the Mississippi Delta, it is an agricultural powerhouse. What an agricultural powerhouse, you get things like pesticides and other contaminants in the air. And if these things are found in the air in rain samples and air samples in the Delta, we can most certainly suspect that if we're bordering the Delta, that some of those contaminants will disperse into our air as well. Some other things are the land use and the urban development that undergoes in this area. For instance, when talking about land use, we have a lot of things that go on in the fall, right? And SEC football is one of those things. Our lab collects air samples on the top of Anderson Hall, and we notice that in the fall there tends to be a higher concentration of black carbon in those air samples. Now, we're not saying correlation equals causation here, but it is funny to note that we do have bigger things happening in the fall. With SEC football, you get a bigger crowd of people, you get fireworks, all these different things that go out into our atmosphere. Urbanization and construction. There have been studies that noted that there's a positive correlation between construction and PM 2.5 presence. And this gets to the idea of justice, because where's the justice for people who live? Where's the justice for people who live right next to areas like this, right? Or the people, like look at this picture, there are people working in this area. Where's the justice for them who are right on these anthropogenic sources of pollution? And to make a long story long, that's where we get the idea of environmental health literacy, right? Which is the knowledge and the empowerment to do the knowledge of environments and the, the knowledge of environmental hazards and the empowerment to do something about it. And as you can see, that's what we ultimately leads to environmental justice. And again, you can see there's a slippery slope before environmental health literacy ultimately leads to injustice. So in order to intervene with that, we have to assess and see what we know in the Lafayette Oxford University community, which is why we conducted a survey to assess behavior and knowledge, as well as having an open space for them to express concerns about chemicals that they might think are affecting their household or somewhere closer in their affected areas in the community. So the ultimate goal was to assess a diverse population and see where we could intervene. And this was just some of our recruitment, the things we did to recruit. We did some tabling outside of the union, used the farmers market, the UM daily email as well. So we had over 200 people do the survey or attempt the survey, but we had 114 qualified responses, which means they met the 70% threshold for completion, or their responses had outstanding identifiers that would knock us out of compliance with the IRB guidelines. And also you can notice that we did have majority of female complete the survey, which is something to think about when developing outreach initiatives. So this is what we ended up getting. We had 20 total assessment questions, but here's 7 that had some rather interesting reported some interesting stuff. For instance, nearly 50% of the people who responded to this specific question regarding lead poisoning in their community, nearly 50% didn't know if there's lead poisoning in their community, which is an opportunity for educational outreach. Something that's very interesting is when we asked the questions about pollution, while 75% of the responders thought pollution was a problem, they felt less empowered when you asked them if they do anything about it in their home. They felt less empowered when you asked them if they could do anything about it in their community. That's a major disparity, especially when looking at the fact that, as I mentioned earlier, most pollutants come from anthropogenic sources, meaning that humans cause these problems, so humans should feel empowered to fix it, right? And this even more interesting is just the fact that nearly 90% of these of the people who participated did think that pollution affected their health in some way. Something else to note here is that nearly 40% of the respondents didn't know whether or not the university did anything or cared anything about the environment. That also includes staff. That includes students. Majority of students who filled out the survey who weren't sure as well. That's going to be a big disconnect, and that can limit community engagement with the university and doing things about our environment. We had a series of behavior questions here, and one of the things I noticed that was interesting, that was 14 to 17% of the people who responded did not know whether or not they were exposing themselves to harmful substances. Right now, 14 to 17% may not sound like a lot, but that's nearly 20 people who don't know if they're exposing themselves to harmful chemicals or people in their households, right? So we could be looking at fatalities over 19 because that doesn't include just the people in the household. And like I find this to be much more concerning than the one person who's quite confident that they're inhaling, car exhaust. Also here, when looking at we found these results to be interesting, that 75% of people who responded, they do use water bottles, which indicates that that's not an area, reusable water bottles, which indicates that's not an area of focus that when we're creating outreach initiatives, we don't have to worry about that, or we can be worried about other things like educational outreach. So, and just some limitations from this study, this was a preliminary study or a pilot study. Some of the things that we thought were limitations were the fact that there was an omission of responses, right? Again, we had 200 people, over 200 people open out the survey, but we only were able to accept 114 to make sure you have quality data to provide for you to report. Unfortunately, when you take out responses, that limits the big picture, and that's what we're trying to get. But for a primary, just like I said, this is a preliminary study, and our initial goal was 50 people, and we were able to get well over that. So we're pretty satisfied. So the social desirability bias, that's when, especially in the context of this survey, when people answer a question that they think is right over what their actual practice is. As depicted here, when this woman asks smoker, you can see that she's visibly not okay with smoking, right? It's not socially accepted. He says, of course not. The same thing happens in the survey, and that can possibly take out the big picture. Also, another thing was university affiliations, like non-university affiliations. Again, we only had 10 people who were not affiliated with the university to actually fill out the survey, meaning that it's kind of hard to see to what extent the actual Lao community contributes to environmental health literacy. Because with non-university affiliates like us now, a lot of people aren't even from this specific area. So where we can go forward with moving with these directives is, of course, getting more non-affiliated people to do our survey, as well as getting these results out there. So other people, so we can compare with other people. There have been surveys done in the Delta specifically in other regions of Mississippi, and we can look at how they're tackling environmental health. Planning educational activities and outreach initiatives, right? Like I said, for instance, lead, they didn't know whether or not lead was an issue in their community. This is a good time for us to show them that there's reports to show you whether or not lead is an issue. We can get water testing kits to show people how to test for water in their communities. So we want to continue to identify groups. And we have data about who specifically answered this from, who's a non-affiliate. And we're thinking about conducting interviews to get more of a raw answer from people about their environmental health risks. And finally, we want to investigate personal concerns. Like I mentioned earlier, we left an open space for people to mention chemicals and substances they were concerned about. One of the most popular of the 114 responses were pesticides, PFAS, and microplastics. I think people with the resources, people with the knowledge, just like I was able to provide with y'all today, we have an ethical and moral obligation to get this information out to people. People with the resources will have an ethical obligation to investigate the presence of pesticides and other substances that can be harming the community. And I think that's where we're going to go. So. Yeah, we're frozen. Yeah, that is my research, and I want to thank everybody who's involved. Thank everyone in the Roper Lab. You all were phenomenal. I appreciate my cohort. It was a pleasure. Absolute pleasure. I look forward to seeing what y'all have in the next coming week.
Thank you so much, Mr. Lewis. So now we will have 5 minutes for question and answer. But first, I would like for Mr. Lewis's mentor to stand, Dr. Courtney Roper. And if there are any members of the research team here, will you please stand? Awesome. Thank you so much. So now we have questions answered. If anyone has a question for Bryant from the audience.
Yes, sir.
Hey, great presentation, Brian.
Thank you.
I'm a political scientist, so I have a question. You mentioned something about an IRD that you had to complete to do the study.
Yes, sir.
What is the IRD?
So it's sorry.
I said, what is an IRD?
Okay, so it's institutional review board that makes sure our study is maintaining the dignity and the privacy of anybody, especially we're using human subjects. Humans filled out our survey. We don't want to do anything that identifies who these people are and exploit their privacy in any way. And the institutional review board makes sure we're staying online with that. Yes, sir. Any other questions? Yes, sir.
I'm curious, if you were to put in an additional question in your questionnaire, which they'd like to expand and give the study over again. What question would you want to put in for example?
The questions that well, the questions that I showed, those were just 7 of 27 questions. There were other questions. Personally, I think we asked a sufficient amount of questions. There were questions asking about smoking. Are you people who might burn garbage in the back of their backyard? Plenty of questions. Something I would definitely do different is wanting to let them know that you can be honest. And that's that social desirability bias. We want the truth. Because knowing the truth, we can do the correct outreach initiatives to ensure environmental justice for everybody. And by justice does mean equal protection, but that's the goal. In order to get equal protection, we need equitable initiatives. So, yes, sir.
How is conducting this research and maybe just the different things that you've been acquiring through seeing the world through the eyes of this research topic change maybe the way you see yourself in the world?
So there was a Dr. Craze. She was a research assistant with Dr. Roper's lab. And when she initially brought this idea to me, I was like, I want to do a survey. I was like, I want to test pesticides. I want to do some wet lab stuff. And when I started doing this, when I started reading the papers, I was like, but it's the ignorance that's leading to global fatalities. As you probably saw from my thing, I aspire to be a physician one day. And it would be ignorant of me to ignore the fact that most people are losing their lives because they simply don't know something. And that's what changed. I think that's where my viewpoint kind of changed. I need to focus on the bigger picture and what people think and what people know rather than what I want to focus on. Yes, sir. Did that answer?
Yeah.
Let's give Bryant another round of applause. Next, we have Mr. Razavier Davis, who is a psychology major here at the University of Mississippi, and his research presentation will be about discovering pathways to restorative justice. Mr. Razavier Davis.
Good morning, everybody. My name is Razavier Davis, and I'm a student at the University of Mississippi. And my mentor's name was Dr. Ayanna Edgman. And today I'll be talking to y'all about discovering pathways to restorative justice. So to start off with a question, what do y'all think would happen if the people that a system or a program was created for didn't have a say in how it was made or what was put into the program? I'll take one answer from the crowd.
Chaos.
Okay, so thank you. Yeah. That is one thing. And there's plenty of different other results that can happen, a lot of which can be negative. A lot of the time when people don't get a say into the program that may be designed for them, it can lead to many issues not being resolved because of the fact that maybe the people that made the program don't understand the roots of the issue, or maybe the people involved might not even be interested in it because of the fact that it's not tailored to exactly how they would normally receive such feedback. And that's exactly why we're conducting this study today. We we want to be able to discover if there are alternate diversionary pathways that help justice involved youth navigate to restorative justice, and if that information can be generalized to not only justice involved youth, but maybe other groups within the country or world. So normally, whenever researchers conduct any type of study or research on human populations, it's all is children more than any other population are normally seen to lack a lot of insight. They don't have that much representation because researchers tend to believe that since they're so young, they might not have as much insight or any input that they can put into the study. But that is far from the truth. Most of the time, children more than any other population can tell you more about themselves or how they will receive such things way better in many other cases. And through helping children be able to express themselves and be able to give researchers the ideas and the perspectives on whatever programs that are made for them or wherever studies are conducted on them, it can help lead to so many different new policies that may actually effectively gain engagement and success from the children population. This goes even more hand in hand with justice involved youth. To put it short, justice involved youth are children who's interacted with the justice system, whether or not it may be through the fact that they had to go to a court case specifically for them, or maybe they've been sent to a juvenile detention center or a community service project because of misconduct. Any youth that's been through the court in any sense of level in pertaining to themselves are usually considered a justice involved youth. And just like normal children, justice involved youth are also extremely underrepresented in research. This research study being one of probably the first research studies conducted on this specific topic. And because of this, justice involved youth tend to actually face many, many barriers throughout their lives, especially those who may have been in the justice system way longer than others. Some of these barriers can include things like the fact that they can't properly assimilate into normal life because they haven't learned any of the job or interviewing tactics that are normally taught in school or college, or the fact that because of the fact that since they've been in the jail system so long, they don't exactly know how to interact with normal people that are not also in the same system with them. And many of these issues end up going untreated because there's no research that actually helps or goes into it. And because of this, there's a high risk of recidivism within the youth population. And recidivism is whenever a youth ends or just anybody commits a crime, there's a chance that they might actually recommit the same crime or commit a different crime later on down the line. And a lot of this seems to maybe stem from, again, that lack of education. A lot of the time, it is even seen that a percentage of justice involved youth who don't get their GED at the very least tend to actually recommit similar crimes later on down their life. But unfortunately, due to the restrictions in this research, it's hard to actually obtain the data as into how deep this goes. Most of the time, not only children, but also prisoners, which justice involved youth usually also constitute as, have a highly difficult chance of being able to share their experiences. And this is usually because of the fact that the institutional review board that are over many of the studies conducted has such tight protection on top of children and people who've been in prison because of the fact that they're all protected groups of people, or groups of people who are at a slightly higher risk of being exploited by researchers. And because of this, much of this type of information that we would need in order for to help them out just isn't available. So the best way to be able to do it is to ask them what are their experiences. Normally, when programs are created for justice involved youth, they end up failing to gain a good amount of engagement or a good amount of positive results. And this is usually because of the fact that they're not tailored to or created with the input of those youth in mind. It is even said by the youth court judge of the Lafayette County area that many of the programs that she tried to use in order for the help justice involved youth out tend to not work because for some reason they don't like the program, whether or not it's how it's made or how it's or maybe some of the things that's going in isn't really tackling the issues that they would like to see tackled. And for those who don't know what the youth court is, it is basically a court system designed specifically for children within specific counties. And there can only be a court if the county is above a 50,000 population. So many counties, especially in Mississippi, that doesn't have a huge population, don't even have a youth court in the first place. So the fact that even the ones that does have a youth court tend to not gain that much engagement from the population that is trying to help is very telling. But studies have shown that programs that are designed with the input of youth in mind have actually seen not only better engagement, but even way more positive results in the end goal, which means that studies like this are very essential to helping the future, the children of the future go forward. To speak about diversionary pathways are simply alternative routes to seeking restorative justice within youth. And normally, a normal pathway that would a child within a youth court would end up following would either be something like community service, or them being sent to a juvenile detention center, or being sent to participate in certain programs that are supposed to help them rehabilitate. But the only issue is that many of these pathways tend to lead to them not really gaining the foresight or the information that they were supposed to gain at first. And studies usually, and that's why this study is trying to help explore unique methods catered specifically to the youth involved. And this comes in the form of, again, restorative justice. Restorative justice is when a person who committed a crime tries to make amends with the victim of their crime, and that in a way helps them rehabilitate because it gives them more power to be able to actually control their situation and realize that, okay, maybe they actually can do better in the future. Maybe they don't have to keep doing the same thing over and over again. Uh-oh. And for the methods in this, starting from that here on out, unfortunately, the study is ongoing. We haven't fully completed it. So most of the stuff now will be spoken from a future tense. But to start, our research is going to include 4 total focus group of people, and each focus group is going to have around 4 to 5 justice involved youth inside of it, with each of them ranging from the ages of 12 to 17. This research study will include 2 undergrad resident assistants and 1 graduate resident assistant, as well as 1 behavioral analysis that is over the entire study. And the 3 resident assistants are simply there to help the person that is over the study move things forward. And the setting will take place in the South Oxford Center. And for each individual session, they will last around 120 minutes each. And each session is going to be a focus group, which is basically an open-ended discussion that's facilitated by someone who's asking questions. And this is in order for the gain engagement from the students and from the children, and for to get them to actually provide us perspectives and thoughts about what they may want to see, or what they may want to may want to see for us, or what they might want to see for the future when it comes to policies and helping them out with their rehabilitation. And the sessions will take place in a neutral setting. This is because of the fact that we want to be able to actually have the youth have a sense of control and not feel like they're being interrogated, or feel like they're being in a place that they're just not very comfortable in. And each setting will have recorded sessions. Oh yeah. Oh, okay, that's not working. We will also we will also give each youth a sheet that will have basic demographic questions for them, allowing them to be able to give us the statistics that they may, that we can be able to use for future studies to be able to help figure out maybe what crimes are associated with which group of people more often, or which age group may be considered to do which crime more often. Okay, there we go. And this is just a list of some of the questions that we may ask. This isn't a list of all of them, but some of the more important questions. And these questions were designed with the sole purpose of helping the youth actually just give their own perspectives. We don't want to really give them any close-ended questions to where it may be yes or no, or any type of questions that may only lead to just one specific type of answer. We're trying to gauge as many different types of answers as possible and gain as much discussion as we possibly can have. So we designed the questions with that in mind. And as stated earlier, the study is still ongoing, but unfortunately, due to the method of which we are conducting the study, we cannot actually have any results as of right now, or even any predicted results. And that is because we're using a process called inductive thematic analysis, which is simply us not wanting to create any predictions in order for to keep from having any type of bias or any type of favoritism towards any type of themes that we may see pop up when we're asking these youth questions. But we did actually have predictions for maybe the possible demographics that we may be meeting. This includes the fact that we believe that most of the cases will be referred to the youth court within the Lafayette County area, or that each of the cases will involve simple assault or disorderly conduct. They will be fairly moderate in the severity of them. That majority of the participants would be male, and that the age range would be more of 14 to 17. And and for discussion, we believe that as of right now, this is the first study on this topic. So because of that, it is extremely unclear as to exactly how this would affect anything. It could have no results whatsoever, or we could even go as high as influence state or country level policies about maybe different programs involving justice involved youth, or even other groups of people who are underrepresented that could seek to benefit from this study. And we believe that through this particular study, that hopefully future research will be done on similar topics, allowing for more engagement from different programs throughout the state and throughout the country. And lastly, for limitations, unfortunately, justice involved youth will only be recruited from the Lafayette County area. Many of the counties around this county does not have a youth court. So because of that, it leads to the fact that there could be way too much variables into maybe how to help justice involved youth out, because youth from a county that may not have a youth court could go extremely through different processes than youth that have been through a youth court. So because of that, we're only really pulling from the Fayette County area, and that also means that we lack a lot of generalization ability in our study, because we will only be pulling youth from one small county instead of from different counties. And lastly, because of the fact that we're pulling research from youth within this county, we wouldn't be able to share perspectives on maybe those youth that are in counties that are lacking a youth court. So because of that, we want to just stick to this particular county. And I just want to thank everybody who has helped me out throughout this process. And if anybody has any questions, I am open to answer. Yeah. Oh, yeah. And I do, sorry, I do want to thank my mentor again, Dr. Anna K. Edgman. Unfortunately, she's not here today because she had to go to a conference yesterday, but she's an absolutely great mentor, and I'm happy for the fact that I had chose her.
Thank you, Razavier. Any questions for Razavier?
Okay.
Dr. Godfrey?
Yeah.
Great presentation. I am question. So I'm looking at your depth for future investigation of matter. Will the study contact the youth group to examine data just in cases?
Unfortunately, because I think my mentor is in constant contact with the person that is within the youth court, but it seems like that information is extremely private. Usually, any type of justice involves statistics among the children population is almost impossible to get. So the chances of us possibly being able to get it are slim to none. But we do plan on actually keeping up with the children after the fact just to see how the processes go and if it's any different than any other cases that we may see.
So, related question. Did you already include these justice involved youth? And if so, what is the racial?
We have not yet. That's one of the reasons as to why the study is still ongoing. We're using a bunch of different methods in gaining the youth, but we only have a very small handful as of right now, and I haven't been informed of what that demographic is just yet.
Dr. Avant?
Some question to your for target populations such as yours. How do you plan to overcome the challenge be in words that you better instead?
Fortunately, the IRB has already improved the study, but the process of making sure that it gets improved gets approved is usually making sure that you have every single thing and every single variable of what could happen already accounted for and planned. Because since children themselves are such a vulnerable population, you have to basically give the IRB an answer to every single question as to, and every single question that they may have, and we were able to do that.
Any other questions for Razavier?
I have a question. I know that the presentation that the study isn't done yet, but what are you looking most forward to for the rest of the process?
For the rest of definitely gaining this perspective of the justice involved youth, because even outside of this study, I want to counsel young children. So being able to actually firsthand see that some of the population in the future that I would be counseling, it would be great to actually see the experiences of them and be able to get a firsthand account as to, some of the issues that may be prevalent.
Absolutely. Thank you.
Let's give Razavier another hand. Next up, we have Mrs. Shakiya Drummond. Shakiya is a biology major from Russ College, and her research deals with the role of early life exposure to hyperglycemia. Shakiya Drummond.
Good morning, everyone. My name is Shakiya Drummond. As y'all know, I'm a biology major. I attend the illustrious Russ College, and my topic was about the effects of early embryo hyperglycemia on the adult function with the insight from a zebrafish model. So, starting off, this is a zebrafish heart. It's a super tiny heart. It has 3 chambers. This is the bulbous, the atrium, and the ventricle. Starting out, my research question was, does early embryo exposure to hyperglycemia affect adult heart structure and function? My hypothesis was early glycemia exposure causes long-term cardiac change. We use this with the zebrafish model. Many people don't even realize that when you're pregnant, that's when the heart starts to form, and whatever you feel, your baby feels. So the stress that you go through, your baby goes through it, and that affects the heart function. It may not form correctly. You might not even know that you have a heart issue until later on in life. And that's what we're basically focusing on is the little tiny heart that starts in the belly, how it forms throughout your life, the adulthood. Very common. It's not talked about enough. That's another reason why I'm very intrigued with this topic. It's not talked about enough. And when you're in the stomach, many pregnant women have diabetes. They have high blood pressure. So when you have high blood pressure as a parent, it goes to your baby, it brings all the glucose, it brings the stress on the baby. You have the heart is infarcted. You may not have enough effect to the baby. And it does disrupt the pathways. It causes the baby to become extra weight that it doesn't need or may not gain weight at all. Study shows that I don't know how to see it, but this is like a normal heart, and this is the right ventricle hypertrophy. You see how this one is normal and this one is a little bit smaller. On to my research. This is the zebrafish model. We use zebrafish because it's transparent, it develops quickly. The zebrafish heart forms within 24 hours, while it takes 9 months to form our heart and to form our whole body. A zebrafish their heart is formed, and the heart is located right here. It's very tiny. Okay, right here, bottom. This is my process. Put 3 groups: glucose, mannitol, and E3. That we're exposed to those 3 groups: glucose, mannitol, and E3. To do this, we took a bucket of ice and we made sure that it wasn't alive anymore. We took the measurements of the fish and we also investigated the heart under the microscope. I made a vertical cut and a horizontal cut and I retrieved the heart. And then I took a picture of the heart on the computer screen under the microscope and I used PFA to store the heart. I stored the heart for 24 hours and then I went back and put it in PBS and I took pictures of the heart again. And I took the necessary measurements to evaluate the heart. These are 3 of the different fish. This is the E3 control fish with the heart. This is the one where I successfully retrieved the lung, the ventricle, and the atrium. This is the glucose heart. I'm going to retrieve the atrium and the ventricle. And this is the mannitol fish, the bulbous, and the atrium. Although I didn't retrieve all 3 changes of olives, there's not much of a difference between them. There's a significant difference in the heart size, ventricle to body ratio. As you can see, they're about all the same. While there was a glucose group being a little bit smaller, there was a large variety across the groups. We had no symptoms. For the future directions, in the future, we hope to analyze the heart a little bit more and go into detail about it, not just seeing like the outer part of it, but also like the inside part, which will contain gel slicing. And also we want to investigate the gene expression changes, and we want to start with bringing awareness to maternal health, cardiac health, and open more doors for conversations of breaking the cycle of heart disease. Early glucose exposure can help shape lifelong heart health. Zebrafish helps us understand effective interventions. Improving maternal health is key to prevention. I would like to thank my mentor and his grad student, Sesmitha. And also I want to thank my cohort. Y'all have been my rock through all of this. I really appreciate y'all. And I want to thank Dr. Howard, Dr. Godfrey, and Mr. Bryan. Thank y'all so much. Oh, and my mentor is Dr. Bloomcats.
Dr. Bloomcats, would you please stand? All right, now we have questions that you may have for Shakiya.
Yes.
Did you enjoy taking the home?
My first time taking it out, it was kind of traumatizing. I didn't like it because I would have to actually cut a fish open. It was definitely scary for me, but with the help of Dr. Bloomcats, he showed me step by step, so I wasn't as scared after he did it a couple of times and then I did it. It was very good. I enjoyed my whole process working in the lab with them because I've never experienced anything like this, so it was definitely a good process. I loved it. Yes. Huh? There. If I can go back to this.
You wasn't wrong, Chris.
Oh, I can just use it here. So they're really, really about 4 inches. They're not even that big. They're really small, and most of them are even smaller than that. So they're not that big. So cutting their heart out was kind of a challenge because it's a tiny little heart. Yes.
But your data said that there did not be a big difference with the size of the heart. Was that what you expected from the octave or?
I really didn't know what to expect if I didn't receive all channels. This is only the ventricle. So I didn't see a difference with the ventricle. Like when I was looking under the microscope, they all look pretty much the same to me. But like when we measured it, when I did the measurements on the computer of shaking, I didn't really see a difference. They were all pretty much the same amount of numbers. It wasn't extremely high and it wasn't extremely low. Not really. Not really, because after I looked at them, I was like, I don't think this, like after I really like looked at them, like it's not gonna really be a difference. They're all the same size. Yes.
So this research has the potential to have a great impact on particularly women who are carrying children. Is there a certain population that has more of a risk, or can this be one type of research that is generalized for a certain population, or do you plan to generalize it?
As far as like it being generalized, the population is high all over because like it's not really talked about enough. Like women don't really get the attention they need when they're pregnant. When pregnancy is like a very like fragile thing, so it's not really talked about enough. But the percentage is high for all. Cardiovascular health kills about 17.9 million a year, so I do think it's gonna be generalized in the future. Yeah, I really wish I had more time to do it, but I'm gonna get to it. Thank you.
Next, we have Mrs. Berlenda Gelin. Mrs. Gelin is a social work major from Alcorn State University. Her research deals with substance use among second-generation Haitian youth, and it is a qualitative review. Ms. Berlenda Gelin.
Good morning. My name is Berlenda Gelin. I'm a second generation second generation Haitian youth social work major at Alcorn State University, coming from New Orleans, Louisiana. And my research topic is substance use among second generation Haitian youth, a qualitative review. And I also like to thank my mentor. I was mentored by Dr. Zane. So for my overview is pretty much what my topic is about, what my research is about. It was based off of 5 peer-reviewed studies. So I didn't have enough time to actually find more studies. And also the studies there wasn't a lot of studies on substance use, particularly in second generation Haitian youth. So in the 4 weeks that I had to find studies, I've only found 5 peer-reviewed studies. And so the themes that I found in those studies were individual vulnerabilities, systematic social cultural influences, acculturation and stigma, and barriers to care and underreported. My goal is to inform more cultural grounded interview. So for my introduction, most youth in the Haitian community struggle with knowing their identity, discrimination, and systematic challenges. My purpose is to identify factors influencing substance use and gaps in research. And my method is narrative and thematic synthesis of peer-reviewed studies. So pretty much I analyzed all of the 5 peer-reviewed studies and in my qualitative review, and I just kind of just grouped them off of the themes, and then also the years was based upon 2005 to 2022. So for my first theme, it is individual level vulnerabilities. Individual level vulnerability refers to youth who are more prone to be individually vulnerable to substance use or depression because of trauma and self-esteem. So for my first source, what was identified was high pathology personality traits linked to substance use. So high pathology is the difficulty in managing relationships and emotions. Pathology is the study of mental illnesses. So in this study, they took youth from the South Florida community and the South Florida and Miami community, and they pretty much put them in a personality trait group, and they wanted to see if they would come up with youth that were prone to mental illness, or if their personalities linked to their mental illnesses. So 30 out of 209 individuals or youth in that South Florida community were prone to high pathology cluster personality trait. So for my second source, it was also in the same community in the South Florida Miami community, and it talked about the study was about the link of PTSD and how it linked to trauma. How trauma or PTSD linked to gang violence or substance use. And even though there was many or little amount of links to it, there was no actual major link to it. And towards the end, they found out there was no resulting link to drug use. So there was a link, but because they had a major gap and they only studied one group, there wasn't actual a big link. So this highlights internal risk factors like personality and trauma history. My second thing, systematic and social cultural influence. Systematic influences are immigrant policy, economic systems, and racism in healthcare, education, and in the justice system. Social cultural influences are in cultures, what there are beliefs or norms in cultures that pretty much limit help seeking. So, like I said, I'm a Haitian American youth, and in the Haitian American culture, they use more of medicines that they that we make or herbs that we use instead of actually going to the doctor. So that's pretty much what I mean. So for my for my third source, it introduces structural racism in the juvenile justice system and affects coping and care access. So this one also in the South Florida community, and they took youth out of, or they used youth out of the juvenile system and their families and gave them coping mechanisms and put them in like therapy and just different and also they did self-reporting tests to try to see what effects or what things was going on in their family and themselves that they didn't have access to. So they gave them the access to see what are they missing, like what care do they need and everything. And so for my fourth source, it talks about the breakdown of traditional support systems like LACO increases vulnerability. So this one is different from all my other sources. It was mainly, it was a qualitative review, but also it was mainly present and talked about in Haiti. And so they talked about the LACO system. It was a community system that was based upon families that was supposed to come together and help the community and the youth to be for the community and the youth to be less likely to simulate to substance use or HIV risks. But because of the system, because of the poor system in Haiti, it ended up failing. And so that's what the study wants to introduce and talk about, the high risk and how can HIV and substance use, how could they decrease the risk. So and all they talk about the discrimination and community disconnection equals the risk of escalation. Pretty much students that do not have the support and them facing discrimination and the structural racism and the breakdown of the traditional systems, they don't have the support, the access to those coping mechanisms to help them to be better. And I haven't said it before, but all of these lead to high HIV risk in the Haitian American community. So for my third theme, it talks about, my third theme is a cultural relation and stigma. This source was conducted in New York, and it pretty much was about Haitians in Haiti and the United States, which was in New York and Florida. And it kind of just sums up and talks more about how Haitians have identity confusion and cultural science. And most of the time, like I said, being Haitian American or just a Haitian and migrating to the United States, you're more prone to not or you want to keep your cultural beliefs and cultural beliefs and what makes you as a Haitian, but because of the policies and the things that Haitians face in immigration here in America, it's hard to not it's hard to actually identify yourself as Haitian or just I can't think of the words right now, but it's okay. But I also wanted to say that one of the quotes that was in the source was most Haitians are called boat people, and they also were called HIV holders. So what I read was Haitian Americans carry the highest. I don't have a lot of statistics. Like I said, there was a gap in the research, so there's not as much of resources or stats that really talk about substance use, but they talk a lot about HIV risk. So they talk about both them as being boat people or HIV holders. And with that, the culture, like their silence, like they don't want to already their culture, they don't want to go and seek mental health. They don't want to go and find help for themselves because they already feel like, okay, well, we have HIV in our culture. We have this, we have that. And then we also don't want to go to the doctor, so let's just not go. And because of that, that limits help seeking. You don't want to get help, and so then they just end up having high risk of HIV, and then we all know what HIV leads to. So also, fear of being identified as Haitian, like I said, hidden help seeking, and then culture stigma around mental health and sexuality leads to denial and silence. Like I was saying about HIV, and in the other source, it talks about the high HIV risk because of the breakdown of the traditional support systems. Because of those support systems don't actually help and stimulate the youth, they're more prone to drinking alcohol, actually submit but substance use and other. Okay, so for my fourth thing is barriers to care and underreporting. So because of all of the things that I listed in the sources, a lot of times Haitian Americans, they mistrust the system. I know me as being a Haitian American, but also being a Black person in America, I don't want to actually, or sometimes I mistrust the system. I don't always actually want the system to, what's the word? It's just something about being Black in America, you know? And also the fear of deportation, language barriers, being Haitian, I don't speak 100% Haitian. So that's one thing that like the identity part, like I claim being Haitian, but also being American, I'm more linked to my American side than my identity of being Haitian. And then also the cultural gaps. Stigma prevents open conversation and service use. So like I said, Haitians actually don't want to have the conversation of the HIV risk, of what's going on in the mental health and everything in their culture. And so data suggests that Haitian youth are often overlooked in research. So a lot of times, if I were to ask you who are Haitian people or what is a Haitian American, a lot of people would not know. Like I've asked somebody, I said, I'm Haitian, and they would say, you mean Asian? I was like, no, Haitian, I'm Haitian. And it was like, oh, I never heard of that. Where is Haiti at? You know? And so because of that, and because of like the lack of like we talk about Black people in America, but there's limited research about that. So think about the immigrants just being in America. There's limited research. Like I could barely find any research about Haitians, second generation Haitian Americans in America, let alone just Haitians. And it only really talked about HIV. Okay, so for my implications and recommendations, I want to promote cultural competence and trauma-informed care. I want to increase research and outreach in immigrant communities. So pretty much, I want more seminars and education, mental health support for Haitian American youth. When I was reading these peer-reviewed studies, I realized how well I was actually educating myself. I'm not really a reader, so McNair really pushed me to deep dive in my culture, and that's what I really wanted to do. But one of the things that really stood out to me, I didn't realize how much. Well, one, I realized, but I didn't actually know how underreported Haitian Americans were. And then also, I didn't know the high risk of HIV risk in Haitians. And so because of that, I feel like there should be more interventions, there should be more seminars going to Haiti, or even in, like I said, only three of my sources were in Florida, and then one was in Haiti, and then another was in New York. But I live in New Orleans, and there's a big population of Haitians in New Orleans. So just think about all of the research or article journals that could be placed in the different type of states or other countries. So also I would like to, I want researchers to look at my research and try to develop cultural rooted interventions, like I talked about, rebuilding the lacrosse system. And that pretty much is in Haiti. Like I said about the system, it fell down. Families, they just gave up. And because of that, it ended badly. Like, it just, yeah. In conclusion, substance use among Haitian youth is shaped by intersecting forces. Pretty much the system, what goes on in what the government, the system, and just the community around them, wherever they go. Urgent need for equity in care and research inclusion, like I already said. Our systems must stop hiding Haitian youth between the cracks. That was in my last source that was conducted in New York, and they had a lot of inspiring quotes about how Haitians were actually in the cracks, or they identified as just American or just Black Americans, just because they wouldn't actually get enough help that they needed if they just said that they were an immigrant or they were Haitian American. And then more studies and cultural competence supports better futures. I believe that if there's more research on Haitian Americans and even immigrants, and putting more putting more research and seminars and book reviews into those communities, can really support them to do better, to be somebody like me trying to pursue my, PhD masters, So I think that would actually help with better futures. And that's it. I would like to thank you. I was so nervous, y'all. Y'all didn't know. Thank you. Do you have any questions? As your mentor. I would like to thank my mentor, Dr. Zhang.
Is he here?
You. Yeah, he's right there.
Yes.
Any questions?
All right.
You mentioned bringing about different substances that were used for tell us some of the substances that you found in literature, that the patient used to either utilize?
Haitian Americans, when they migrate, I know there's a low rate of marijuana in Haiti, but when they migrate when they migrate into the United States, it's a way like bigger scale, if that makes sense. Like when they go into New York or Florida, being in a black community, you have more access to marijuana. So that was one of the top things that were introduced into the sources. Well, one of the sources. Thank you. Any other questions for Berlenda?
I know that you said that it was not a lot of research, which can be frustrating when you're trying to do things. But also it's a good thing because it means it's a lot of room for you to do work. So what do you think you want to do next with this?
I would like to actually, okay, so when we went to the conference in ATL, I seen a lot of Jamaicans or other, I guess, other Americans, other nationalities that talked about pretty much around the same things that I talked about, but that it was different. So I want to actually elaborate more and conduct interviews and other things. Like I said, there's limited research, and they're all only conducted in Haiti, New York, and Florida, because that's where Haitians are more prone to. If you don't know where Haiti is at, Haiti is pretty much like near Miami, Florida. So it's easy for them to just get on the boat and just get there. And then after that, they just leave and they go to New York. Okay. So I want to actually conduct research maybe like in New Orleans, since I live there, or in Louisiana, and see if they actually have the high risk of substance use or what's going on in that area, just because there's just this is very unreported. So yeah.
Thank you.
Let's give Berlenda another round of applause. Moving right along. Our next presenter is Mrs. Charm Hunter. Charm is an allied health pre-medicine major here at the University of Mississippi, and her research deals with understanding microbial behavior using genomic DNA sequencing. Mrs. Charm Hunter.
Good morning, you guys. I said good morning. My name is Charm Hunter, and today I'll be talking about understanding microbial behavior using genomic DNA sequencing. I'm a rising junior here at the University of Mississippi, and I plan on going to med school and becoming a physician one day. So, microorganisms are found everywhere, in deep seas, deserts, desiccated environments, and they are in places that we don't even think they would be. The understandings of these microbes are very important because they contribute to our lives both positively and negatively. For example, one positive way is that they use nutrient cycling, and one negative way that's very familiar is that they cause infection and disease. This leads me into Dr. Cockrell's 500-year project. This project is led on by Dr. Charles Cockrell, but I added my own twist to it. This 500-year project is the main objective is to study and understand the effects of microbes when they go under long-term desiccation. As you can see, these two vials right here, they include Quercydiosis and Bacillus subtilis. Quercydiosis is a cyanobacterium, and Bacillus subtilis is an endospore bacterium. These vials were then put into this box on the day of July 1st, 2014, and this box symbolizes a very, very dry and very low nutrient, no water environment for these microbes. On the day of June 30th, 2514, this is the time that these microbes would be expected to come out of this box. But Charles Tocqueville did his follow-ups on these microbes, and he found that they were impure or contaminated, which is this arrow right here. And these microbes were growing upon the cyanobacterium. You're probably like, Charm, why does this even matter? Although this project is led on by Dr. Cockwell, I would like to further this study and apply it to hospital settings. By sequencing DNA and revealing potential genes of these microorganisms, we will be able to find out, we'll be able to help staff of hospitals and also people who study microbes survive under disinfectant and sterile techniques. This leads me to my research question. What genes help bacteria survive in dry, nutrient-limited, and disinfected environments? Methods. So, the Sphingomonas melonis, Bacillus subtilis, and methylocorticum was all extracted by Dr. Cockwell himself and sent to the MESA lab for sequencing. Methods. As you can see right here, this is me uploading each organism into this pipette using a rapid barcoding kit. This was extracted into this flow cell and then was clipped upon this grid ion and ran for a total of 48 hours for accurate sequencing. Once the sequencing is done in 2 days, we then use the data analysis bioinformatics tool EDGE to complete a genome assembly and genome annotation. So you're probably like, what do we get in EDGE? Why is this even a thing, and why do we even have to put these organisms within this program? We get things such as KDE plots. KDE plots show the overall accuracy and quality of our organisms. As you can see, this is methylocorticum, and the more that it is up and right, we see that the more that it is up and right, that means that it is more accurate. When we have accurate reads, we can know if we want to use this data or not, or if we just want to overall keep trying over and over again with our sequencing. Our genome assembly. Why are genome assemblies important? As you can see here, Methylbacterium aquaticum, Sphingomonas melonis, they both have a complete circular genome and extra DNA plasmids. These are the plasmids here. But in Bacillus subtilis, it has one circular genome, but it has this little bit of a rod here. This little bit of rod symbolizes this.
With the 3 dots, under more, and hide. No, isolated.
You get this. Oh, yeah.
So these are our genome assemblies. As you can see, methylocorticum has a complete circular genome, well, almost complete, with plasmids and extracellular DNA, along with Sphingomonas melonis. Now, Bacillus subtilis, we got one contig. But right here, we found that it has a little bit of rod. This means that this organism needs further work and it needs to resolve the genome in one section. But what can this genome tell us? This genome tells us our overall results. So as you can see, I highlighted some genes that are very relevant to microbe survival.
The first gene is DNA K or GRO-E or GRIP-E. This gene was found in all organisms. This is a chaperone gene. A chaperone gene is basically a protein that shapes organisms back into their original form when they go under harsh conditions or they go under overall stress. The REC-A gene, which is also found in all, which is a DNA repairer. So when organisms go under a lot of stress or in harsh conditions, their DNA is sometimes damaged. So this means that if organisms have this type of gene and they live on a hospital surface and we keep disinfecting it and disinfecting it and disinfecting it, but their genes are destroyed, but they have this gene to keep repairing and repairing. That means that this gene is more than likely going to live a long time on a surface. STIN, which is found in Spingomonas melonis, it is a citrate transporter, a nutrient uptake and metabolic survival under harsh conditions. So when I think about this, I think about an animal going into hibernation. They take in so much energy or so much food and they go into hibernation for a long, long time. And they only use this energy when they need it. So this is kind of what this gene does as well. It takes in so much nutrients and metabolism, and it only releases it when it needs it. Then SPU-M and SPU-A, which is found in Bacillus subtilis. Previous in the presentation, I told you that B. subtilis is a spore-forming bacterium, which is one of the most protective strategies of survival when it comes to desiccation. This gene is a sporulation signaling gene, which initiates sporulation of a cascade of events. Connections to hospital. Okay, let's go quick. Some of these genes that we found were in the most popular infection bacterial disease in hospital, which was Pseudomonas aeruginosa. And also the biofilm microbes, they live on hospital sinks and equipment. Environmental survival genes may help explain hospital persistence. Acknowledgements. I would like to give a heartfelt thank you to Dr. Bex Prescott, Dr. Charles Cockwell, and the entire MISA lab. If you would like to ask me any question, my email is on there. Thank you so much. But you can't go down yet. Is your faculty mentor here? Okay. Does anyone have any questions for Charm? Yes, I really did enjoy my experience. Overall, I do want to become a doctor in a sterile environment, such as a surgeon setting. So I feel like if we figure out what these microbes are doing gene-wise, we can potentially put this into a hospital setting and find out how settings can be more sterile. Yeah. You can go ahead. The gene, yes, overall, the genes that I expected, there are way more genes within my research paper, but these are just the genes that were very interesting to me. So the DNA damage repair gene is one that I really expected to come out of the genome. Any other questions? Let's give Charm another round. We have Mr. Bryson Boone. Mr. Boone is a criminal justice major from Russ College, and his research deals with factors influencing police criminal misconduct in Tennessee and examination of officers arrested between 2005 and 2011. Mr. Bryson Boone.
Good morning, everybody. How y'all doing?
Usually police officers are seen as a useful tool in our communities. In recent years, there have been a rise in police misconduct in various states in the United States. Many people believe in the community believe that it's a current issue. I made it my mission to analyze the years from 2005 to 2011 to elaborate on the facts that police misconduct isn't as new as many think. From the National Police Report, I noticed the police commit various different crimes, but the main two I seen was sex crimes and drug-related crimes. From my source, Stinson detailed that if an officer had a prior conviction, it most likely means the officer will commit a crime once again, leading to a cycle. From my source, Skolnick, it detailed the action that police to do tend to protect one another when vouching for one another using the code of silence type tactic. Lastly, from my mentor, he and his colleagues discussed that there are various different factors that influence actions of the police, and which I'll simply put as age, ranking, and gender, but I'll plan to elaborate on those shortly. The nature of my research was to is explain that police misconduct wasn't a current issue and also examine the factors of what crimes and why they commit said crimes. The sample consisted of 233 officers convicted in the state of Tennessee. I plan to break down and analyze percentages of the two categories of crime. The data plan was rather descriptive, even though that we had two dependent variables being displayed at the time. The two dependent variables I just mentioned were sex and drug-related crimes. The factors I had mentioned earlier, such as the age and the rank of the officer, were the independent variables that we tried to correlate with predictability. The predictability is what we use to see how common the crimes will occur and see if it has a common goal, if the officers would do it again or so. This table contains all the collected data from the various tables we used in my paper. Roughly the age of the officers were 37 years old, with 94% were male. More than 60% were off duty or wasn't working a case while committing misconduct, while 40% was working. Less drug-related crimes occurred from the higher-ranking officers compared to the low-ranking deputies, along with having 10 years of service under their belt. The findings from the previous slide and what I detailed was that not all the independent variables impacted both, such as tenure, duty status, and rank played a pivotal role in drug-related crimes. For example, a lieutenant wouldn't commit a drug-related crime, while with sex crimes, the main factors were rank and also gender, such as high-ranking male officers will commit those crimes against low-ranking females. With the findings from the previous slide, you'll overall see that some variables were more important or outweighing one crime than the other. Along with that, you could see that sex crimes were more stereotypical and along with factual in the point of view how men will commit those said crimes than females. The limits to this study was that the sample consisted only from Tennessee out of the other 49 United States. It also didn't contribute to the other said crimes that they had committed, like I mentioned in the intro. To conclude on my research, the two crimes committed by police were drug and sex crimes, in which position and tenure are the key reasons they do such. It is mainly due to the lack of accountability being on their point as they climb the positional ladder. Overall, I will say that even though people believe that police misconduct being a current issue, it was a current issue back then. Thank you. I'll open up for the questions. Now let's thank my mentor, Dr. Bakting in the back. Yeah.
Does anyone have any questions for Bryson?
Well, what I'd say that would change is just having a rotation of like how the duties of the officer instead of just having them in one department for the whole 9 or 10 years of their service. Yes, ma'am. That's correct. I'll say more than 50%.
What implications does this have on society at large?
I'll say like overall at large is more that the officers would be more like people trying to get more well with the current state of how people trying to defund the police and all that is contribute to that ideology instead of like building more trust with the community.
Any other questions? Let's give Bryson another. We have Mrs. Jamiria Massie, and it's misspelled. It is misspelled on the paper. I didn't do it, Mr. Brown. Jamiria is a psychology major here at the University of Mississippi, and her research deals with deliberate practice and the usage in therapeutic settings. I present to you Miss Jamiria Massie.
Good morning. How is everyone doing? That's good. There you go. Okay. So my research deals with deliberate practice in the process of learning psychotherapy. And I was mentored by the wonderful Dr. John Young with the help of his graduate students. So the agenda for today, I will be doing an introduction, talking about cognitive behavioral therapy, not deliberate, therapy, competence, and feedback, then deliberate practice. Then I'll do a hypothesis, methods, procedures, followed by the results, and in my conclusion, which will go in detail about my discussion and limitation. So my introduction. So what is cognitive behavioral therapy? Some of y'all may know it more as CBT, and it's one of the most well-known types of psychotherapy. And this guides the patients to stop avoiding uncomfortable experiences while also targeting the patient's thinking patterns that are causing distressing emotions. So this helps navigate social, professional, personal situations and more productively with less distress. So competence in therapy. Therapist competence is applying interventions in a useful manner with attention to individual context. So this is a very complex skill, and feedback is absolutely necessary to develop competency. However, it's very hard to obtain. The reason why, it takes a lot of time and a lot of money that insurance will not cover for it. So tools like Listen.io will make it more accessible. So what exactly is Listen.io? This. I will go more detail about this in a moment. So, as we all know it, AI is involved in all of our lives, whether we like it or not. Well, why not use it for something more productive, like helping giving therapists feedback? So, Listen is an AI tool that is used to give feedback to therapists immediately after the session. So, how it works, it will go in the talk time, questions, and reflections. So, I'll explain that in just a minute. So, for example, the talk time. When you listen your video of a session, it will tell you how much you talk. So the practitioner with dark purple, how much the practitioner talked. The light purple will tell you how much the patient talked. And then for questions, it asks you how many open or closed-ended questions you did during the session. So open questions mean more like Socratic, which basically means the practitioner is using these questions in a more broader term. That way it's the patient's responsibility to interpret it and guide it in their way. Closed-ended questions is basically when a practitioner uses more specific terms and words that helps a patient guide them to their answer. So you typically want to use more open questions because it makes the patient be more in charge of the session. And so this right here, listen will show your CT competency. So basically using the CTRS or cognitive therapy rating scale, it will show you your level of competency used in the session. So understanding, pacing, interpersonal feedback, and collaboration. It will show you everything with just your video. It gives you automatic feedback. And it also shows the agenda, homework, guided discovery, and other things. So what is deliberate practice? Well, deliberate practice is used in other fields of therapy, like music or sports. Well, this is individualized training exercise designed by expert and focused and repetitive and monotonic benefits. So monotonic basically means improvement depends on time. So example would be a football player. Well, every football player wants to get better at what they do. Well, what if this certain football player wants to get better at their hiking skills? Well, they can easily just practice over and over again, but instead with deliberate practice, they will go to their football coach, and the football coach will give them a certain type of exercise to do over and over, and that football coach will give them feedback. That is deliberate practice, which is applicable to psychotherapy skill development. So my hypothesis, I actually have two. So receiving feedback will lead to an increased competence. So moderate increase through AI feedback, but also stronger increase through AI plus human feedback and engaging in deliberate practice exercises. My other hypothesis was that therapist speaking time would reduce as competence improves. So my methods. I role played as a therapist, and a graduate student from Dr. Young's SITH lab or Dr. Young himself acted as a patient using a case description. So this particular case description was a regular college student who was really depressed, upset, and just had a hard time adjusting to the college life. This student was very involved in high school, and now they're not so involved in college. So they came to a clinic and wanted to get more help with how they think, how they feel, and their physiological sensations. So I used the treatment from well-known CBT manual, the Unified Protocol. So the Unified Protocol is basically modules breaking down that targets your thinking patterns, physiological or physical sensations, and behaviors. So there was 2 successful rounds of practicing the entire manual from start to finish. First round was only AI feedback. Then the second round was AI feedback, but immediately after the session was over, I got human feedback. And then I engaged in a deliberate practice exercise. And then the cognitive therapy rating scale scores track with each session through listen. My procedures. So prior to each session, the patient or whoever was role-playing as the patient will complete the DASS-21. So the DASS-21 is a scale that measures depression, anxiety, and stress. Each session was a review of this DASS-21, discussion of previous homework, teaching a new cognitive behavioral skill, and an assignment of a new practice activity. So upon completion, I took notes through the session on how this session went, in my opinion. So there are times where I thought the session went really good, so I'll write that down. Or there are times where I feel like it could have been better in certain aspects. And then the session was recorded and uploaded to listen by Dr. Young himself. And this gave me feedback to guide improvement. That was round 1. This is round 2. Round 2 is the same as round 1, but instead, I also got additional feedback from discussion with the human after each session. So either the graduate student or Dr. Young himself will give me feedback on how the session could have went or where I did good. Then we transitioned immediately into deliberate practice exercises. So I was guided through it before trying myself. So an example would be, I may have needed a little work on my Socratic questionings or open questionings. So I was told to explain a story using only Socratic or open questioning or open words. That way, when I'm talking to the patient, they can interpret themselves. And this was focused exercise to improve specific aspects of the skill. My results. So I'm going to explain what this is. So this was round 1 talk time. So I started a little bit above 75%, which is really high. You don't want to be that high when you're talking with the patient. You really want to let the patient guide a session. Well, at the end of round 1, which reminds you, it was just AI feedback, I ended up a little bit around 55%, which I'm talking. Then with round 2 talk time, which was AI plus human feedback plus deliberate practice, I started out a little bit below 75%, and I ended a little bit above 50%. My CTRS, or Cognitive Therapy Rating Scale score. So I started out with day 1 a little bit below 40 for my score, and then I also ended a little bit above or nearby 40 for my score for round 1, which is just AI feedback. Round 2 with AI plus human feedback plus deliberate practice, I had a score of above 40. And then I also, at the end of it, had a score that was near 45. So what does that tell me? Well, my competency did increase. It increased more with AI plus human feedback and deliberate practice, and my speaking time did decrease, which lets the patient guide the sessions, which is a really key component in CBT skill development. So these lines can be applied to other practitioners in the field. Feedback is useful for skill development. You need that in order to grow as a therapist practitioner. But it does not always have to be resource intensive, meaning AI by itself is just perfect enough. And this really helps to receive feedback because it could result in openness to lifelong improvement efforts and higher quality of services. My limitations. So there was only one person in the sample size, me. And the researcher was also me, which indicates potential bias. So and also there was a limited time frame available to complete the project. Typically for this type of project, you will have longer than 4 weeks, but we only have 4 weeks to do it. So I ideally would like to spend more time learning basic techniques and longer periods of time to do my deliberate practice exercises. So future research should incorporate multiple participants to allow for comparisons across individuals and improve generalizability. And there is a need for extension of study duration and reduction of external time constraints. Thank you. I also would like to thank Dr. Young and his graduate students because they have helped me tremendously through this process.
Is he here?
Yes.
Okay. Dr. Young is on the Zoom link.
Any questions? Yes.
So, I think it would be very respectful to bring AI into the field. One question, how do you think you should manage the information that you pull from these talks and these conversations confidentially? One, and then 2, what do you think? How do you think patients could be brought to, like, hey, I'm recording this conversation? Explaining open to what you're doing.
So to combine those 2 questions into one, transparency is very key to this. So you want to let the patient know immediately from the start that if you want to increase your competency, I will have to record this session. So usually with an AI, you don't, for this one, listen, you don't have to have the facial, you have to just have the voice, I think. But you want to be transparent and you want to ask the patient for their permission because that's important. It's their session, they're there to get help. So if long as the patient's okay with it, then that should be fine. But how to navigate the information you're getting from it, basically it is to help you see where you are with your learning process because learning never stops. So there were times where when I looked at the score, I was really high in my score, but the next session I was really low. I had ways to improve. I knew where to improve. I knew, hey, let me quit asking all these closed-ended questions. Let me start asking more open-ended questions. Let me talk less. Let me let the patient guide the session because it's their session. Does that make sense?
Any more questions? Yes.
So I was reading something yesterday about AI and people using AI as a tool for counseling themselves. Some were using it in conjunction with the therapy, some people are trying to use it experimentally. And so a lot of them kind of similar to yours. How do you, who's controlling the software? How do you know that your practices are not being downloaded and systems to eventually replace you as a therapist and work with technology? So the thing that is helping you in the end replace you by taking your information to
And that is a very good concept. So this software was arranged by multiple leaders who want to have a more software that's tailored to therapy. So with this software, it can't replace the actual practitioner. It only shows you your levels of competency and also your talking time and all that. It doesn't show you, well, hey, you're depressed, do da da da. It can't replace a therapist. The thing with AI, people try to use it for therapy aspect because yes, it's more available and it doesn't cost anything. But as we all know, in order to tailor to your needs, you're going to need a human to talk to about certain aspects. AI cannot tell you how to deal with the depressive episode or an anxiety episode. It can't do that. It may try to, but it's so monotone. It's not a human. We're humans. We crave other humans. We need other humans to help us through stuff, not just AI itself.
So I would like to see you do more of that when you do your research and your thesis, because that information can be stored. You have to be really careful about it. And this thing, and our collaborator talked about the fact that it actually does a good job responding to human needs. It just can't diagnose things. And so humans are really enjoying it. So I would like to see you do it. Have you thought about it?
For my research?
Yeah.
So I actually talked with Dr. Young and I have decided to join his lab and I will be collaborating with him for my senior thesis.
Okay.
Any more questions? Dr. Godfrey, you had your hand up?
Well, since we learned about R&D, and since you were collecting data for yourself, did you submit an R&D?
So for this research purposes, I did not have to do IRB, but if I was to use actual regular civilians, then I would have to do IRB. Any more questions? Well, thank you.
Hello and good morning. My name is Deshaun Moody. I'm a student at Alcorn State University, the Department of Agriculture. And today I'll be presenting on the diversity of seed-associated microbes in rice and their potential role in supporting agriculture in Mississippi. I'd also like to give a quick shout out to my mentors, Dr. Eric Holm and research PhD student Noah Colonzo. We all know what microbes are. They are everywhere and in everything, and they influence nearly all living systems, that including plants. This project examines the microbial communities associated with rice seeds. Our goal being to understand how seed-associated microbes influence rice health, development, and sustainability. Our approach being sampling, DNA extraction, sequencing, and microbial analysis. You all might be asking yourselves why it matters. Well, we have an ever-growing global population, and with that growing population comes an ever greater food demand. So, to that end, we need more sustainable agriculture. We also have climate change stressing out our crops, causing a delay in their normal life cycle and a delay in when the farmers can plant them. Now, you all might still be asking yourselves why it matters. Well, Seed-associated microbes might offer a natural solution to these problems. Questions I asked myself during this research. Why do microbes inhabit plants? Well, that's for two reasons. Shelter and nutrients. The plant offers the microbe a stable environment for it to live and grow. And the plant also has water, sugar, and nutrients that the microbe can feed off of. And two, protection from external stress, such as environmental hazards or predators, or simply being blown away by the wind. Question 2. What functions do microbes serve? They can help the plant grow, protect it from disease, help the plant take in more nutrients, or even endow a certain level of stress tolerance upon the plant. Question 3. Can these traits be applied to other crops? The answer is yes. Microbial incumbents have already been in use in other crops such as maize and wheat. There's also genetic engineering and genetic breeding where we can help the crops better be able to recruit beneficial microbes. And there's the simple method that's not so simple of transferring bacterial microbes from one plant to another. Why focus on rice? Well, rice is a staple food for over half the world's population. So what better crop to use than one that feeds over half the world's population? Rice also has an incredible resilience, has a high yield to loss ratio, and we think this might be because of the microbes that it has inside of it. So understanding the rice microbial partners might benefit global food security in the long run. To do this research, we had to take samples of rice grains. These samples we got from a rice farm out in the Delta in Sumner, Mississippi. We gathered 5 different varieties of rice for this research, that being Basmati brown rice, sushi rice, black rice, red rice, and jasmine rice. Quick story time before moving on. While at this farm, we encountered one of the why it matters from earlier. The farmer had been having issues with planting his rice grains, and this was because the climate just wasn't right for the crop yet. So he had to set back his time, which delays production. So if we can continue to investigate these microbes, we can possibly help him still be able to plant on time, even though the climate isn't just right. On to the research. Now, before we started doing any analysis, we had to first prep the samples. This meant keeping them at a cool -80 degrees Celsius until they were used, and then when we were ready to do the prep, we had to dehusk the rice grains and surface disinfect them so that we eliminated any microbes that weren't from the rice. They were then grounded up in liquid nitrogen, and DNA was extracted using a nucleosol kit. The DNA is then put through the PCR process. PCR amplifies regions within the DNA, specifically the fingerprint region. This region is called the fingerprint region because it's very specific. It hosts the code of each type of microbe. So each one is different. And through this region, you can tell which microbe is which species of microbe it is. We use primers for amplification of the region, that being 1F LR3 for fungal, 27F 149 2R for bacterial. The DNA is then sequenced using a nanopure grid ion and analyzed using ONT epidemy software. From our DNA analysis, we found that in our bacterial findings, the bacteria that came up as a match most were Olisteoma SD with the highest, followed by Oceanocilla actinis and 3 others. If you look here at the chart, at the bottom blue, you can see Olisteoma SD with a good chunk of the chart, and the other 3 following in space. For our fungal findings, we found that one genera, Farisium, showed up 42% of the time. The species within that genera being Farisium equiseti, Farisium humili, and Farisium hananis. The fungal species that we saw most afterwards were Curvularia soli, Alternaria delicondium. I apologize for butchering these words. If you look at the chart here, you'll see in the blue at the bottom we have Farisium SD, and then the green slightly above it, we have another of the Farisium group, Farisium hananis. Through analysis of the microbes, we found that these microbes fall into one of 3 categories in ways that they could help out plants. That being stress tolerance, plant growth, and nitrogen fixation. For stress tolerance, we have Oceanocilla actinis and Curvularia soli. Nitrogen fixation has Olisteoma SD, the leading bacterial in our bacterial findings. And for plant growth, we have Farisium, the Farisium genera. Overall, our research has shown us that Mississippi grown rice hosts a diverse community of both bacterial and fungal, with an important agricultural potential. These microbes having benefits such as stress tolerance, nitrogen fixation, and plant growth show us that with their help, we could better our agricultural system and find more sustainable agriculture at a lower cost and at a natural cost. That concludes my presentation.
So my first source, done in 2015, it was a study done to examine the different types of belongings, whether that's family belonging, peer belonging, or academic institution belonging, and how it relates to suicide ideation. And they found that family belonging was more significant than the other ones. In my second source in 2023, my bad, it was a study done to see how burdensomeness and belongingness relate to suicide ideation, and they wanted to see whether both of those either mediate, which is change, or mediate, which is help, or moderate, which is to change the relationship. And they found that both more so mediate their relationship. Assistance. Oh, I got it. Okay. Yeah. My next one is perceived burdensomeness. Burdensomeness is feeling as if you are a burden or feeling like your life negatively affects the ones around you. So my first source in 2010, Van, he founded that suicidal behaviors arise from burdensomeness and belongingness. And he also found that when it's experienced with hopelessness, that the desire to commit suicide was higher. And he also found that burdensomeness was more strongly and consistently linked to suicide ideation. In my second source, they wanted to see how burdensomeness relates to suicide ideation in older adults. And they also wanted to see did gender play a role in it. They found that gender didn't have no role in it, and burdensomeness was a very strong predictor. Hopelessness is the belief that one's life just cannot improve. This was not originally part of his theory, but was founded by Dr. Aaron Beck, and he created the Beck's hopelessness scale. So in my first source, the study was done to basically see how depression and hopelessness can predict suicide ideations, future deaths, and attempts. And they found that they both have their own predictive value. So you can either be depressed or hopeless, or you could be both at the same time. And he also found that hopelessness and depression are not just standalone predictors, meaning that they can help and they can predict, but it's not like a really, really strong factor that could actually see whichever it is. Because you can be depressed and like not actively want to harm yourself. And then in the second source, this was a study done on adolescents who have already been diagnosed with depression, and they wanted to see how hopelessness predicts the suicide ideation. And they found that it was very significant. So my purpose of this study was to basically just explore and get more insight about how all three of these factors work together. There are very few studies done on all three. There are studies done on the interpersonal theory with burdensomeness and belongingness, and there are studies done on hopelessness, but not a lot with all of them together. And the aim for this was to just bring more awareness to the social issue and to just have more prevention strategies. So in my methodology, there was a study. So they wanted 165 students from University of Mississippi to take a survey based off of the Beck's hopelessness scale and the interpersonal needs questionnaire. So the Beck's hopelessness scale was just 20 questions based off true or false. and they would ask questions like, I might as well give up because I can't make things better for myself. So they would answer those true or false, or it would be the interpersonal needs questionnaire, which would ask questions along the lines of, well, it was a statement, and they would either pick from like the scale from 1 being not true to like 7 being very true, and that would have statements like, everyone's life would be better if I was gone. So they did a self-survey for like 30 minutes to receive a $10 Amazon gift card, and the results gave us a correlation and an analysis, a regression analysis, and a mediation analysis. But into my results, there was 87% female, 10% male, and then 1% non-binary or prefer not to say. 65% of them were white, 24% of them were African Americans, and then 9% were Hispanic, Latino, Asian, or 2 or more. 21% were married or divorced, and then 78% were single. And then I did an error bar graph to kind of show the range. Okay, so the line represents the range of responses that they had. And so the longer the line means that the responses were more spread out, and then the shorter the line means that they were more consistent. Suicide ideation had its own little box because there was a question asking how, like, were they experience suicide ideation? So 18% of them were, and that's around 30 of those people. So the littler line means that they were more consistent, and people more consistent. And then in burdensome and belongingness, the answers were more spread out. My correlation graph was done just to see how they work together. So hopelessness and burdensome was the highest one, and then hopelessness and suicide to the very public. My coefficient, this is my regression graph. So they did the regression to see which of these factors actually predict suicide ideation. And hopelessness didn't really do well, even though it was tested and it was shown in all of the other tests. When it did the ones to predict which ones actually did, it didn't do as well. Burdensomeness was more significant. So because of hopelessness not doing really well, they had to do a mediation analysis. And the mediation analysis is the mediation analysis isn't a comparison, but it's just to see the direct pathway. So that's why hopelessness has a 0, because it wasn't directly predicted. But burdensomeness and belongingness kind of scored around the same. Some of my implications. So I've had put to create belonging focused incentives. So that could be like more incentives to create a sense of belonging, whether that be for different groups of people. So it could be like clubs for minorities, or just like little things like that. And then tools to reduce burdensomeness, which is financial aid or academic support. And I thought that they can do like scholarships, like essay scholarships to people who might be dealing with mental health issues, that they can like do their essay and then get financial aid because of that. And then also offer mental health services that address all 3. I also feel like professors and people who have strong influence around the students, because they're always with us, should have trainings to where they're more aware of the things and signs so that they could see and report if anything is off with their students. So some limitations on the study was that it was self-reported data. So people could not answer 100% truthfully, which can kind of mess up the results. And then it was a more female dominated sample. It was, yeah, that there was more like male answers, it probably would have changed. And it was just a limited sample overall because it was just for the students that attend the University of Mississippi. So if it was a sample that was done with more spread out over like the region or the state, I think the results would have been more interesting. So in conclusion, we see that suicide is multifaceted, and it's very complex. There's a lot of things that go into it. And then we see that all three of those factors do indeed work together. And if we better understand their relationships, we can create better support systems for our students. And then some prevention would just be to create sense of belonging, purpose, and to instill more hope into our students. That's
Yes, very good.
questions.
And introduce your mentor. Is your mentor here?
So is Dr. Kim in the back.
Yeah, very good job, Ayanna. Yes, go ahead.
My question is just, what have you learned about really that you personally have learned while doing this concept?
The most valuable thing is just more understanding in general. I do think that I want to pursue a career in mental health. So yeah, I think getting more insight about that will help me in the future.
Is there any more like, so you know how you talked about like the three factors, like, is there any more factors that you know to like suicide?
I think, like I said, it's very complex. Like it could be anxiety or little just things like that. Like we just looked at one person's theory because he was focused more in suicide. So yeah, they're not that I know off of my head, but there are probably like a million more that answers your question. It's
Do you think you would want to further your research on hopelessness as soon as like less searching?
Yes, I did read an article. One of the articles did say that it was not a strong predictor, but I also do feel like that was the case because in Joyner's theory, the capacity for suicide, to commit suicide, you actually have to, you have to have the capacity for it. Like you can't be afraid of death or to harm yourself. So I feel like with hopelessness, it kind of like not cancels that out, but they might feel like they're so hopeless to where they just can't physically harm themselves.
If in future studies, if you would like to kind of explore this topic again, would you ever be interested in using maybe more of an interview style line of question or for the kind of make up for some of the limitations with self-reporting?
Yes, I would. Probably like with people who try to commit or know people who have committed or tried to commit. I also would like to further the study and kind of like compare for like different backgrounds. That makes sense. Anything else? Okay, thank you.
Very good job. Next up, we have Kalasia Robinson from West Point, Mississippi. She is a biology major from the historic Tougaloo College, where the moss hangs low and the eagles fly high. And her topic is organic acid testing as an assessment for stuttering. So let's give Kalasia a hand.
Good morning. I am Kalasia, a junior biology major at Tulu College. I had the pleasure of working with Dr. Snyder, and today I will present organic acid testing as an assessment for stuttering. By a show of hands, how many people have actually stuttered in their life? It's nothing to be ashamed about. So stuttering is currently viewed as a speech fluency disorder. So it manifests different ways. You have repetition, where it's like stuttering, where they repeat a letter over and over. You have prolongation, where it's like prolongation, where they take a long time to pronounce a syllable. And you also have inaudible posture fixation, also known as blocking. Is when their body postures to say the word, but no sound comes out. And people also think that it's caused by nervousness, but it's not. So they currently test people for stuttering by having clinical interviews, and as you talk, they will count the amount of times you stutter, how long you stutter. They'll do a case history where they ask family members, friends, educators, employees about how is your stuttering. They also do a quality of life survey to where they ask you questions about, are you embarrassed by your stuttering? Are you most likely to initiate a conversation? Do you think people notice your stuttering? So some of the current treatment options are fluency shaping. Fluency shaping in simple terms are considered to help people talk more easily, but actually it's to replace the disfluent speech with more fluent speech. And they also have therapy for behavior, social, and emotional aspects of stuttering. So persistent development stuttering is actually a central nervous system disorder, which is your brain and your spinal cord, and it's linked to more symptoms besides speech, such as insomnia. So what causes persistent development stuttering? You have neurological and genetic mutations such as NAGPA, GNP, TAB, and turn it off. Okay. And GNPTAG. You also have autoimmune responses such as long COVID. You have migraine, severe forms of OCD, strep throat, neurological traumas such as concussions and strokes, and medicines. So imagine taking medicine to get better and get rid of one disorder and you develop another one. So some common medicines are Adderall, which treat ADHD. Symptoms appear 2 weeks. Metamine treat autism. Immediately after the dose change, symptoms appear. You have phentoin, which treats seizures, and they start immediately. Then Topamax, which treat migraines and appear 1 to 2 weeks after starting. So because the traditional assessments miss underlying biochemical imbalances, the stuttering population took it upon themselves to find their own treatments. So one thing they have resorted to was vitamin B, which is also known as thiamine. And if we was to do organic acid testing from the beginning, they can detect this. Organic acid testing is a non-invasive procedure because it's a urinary analysis. It's easy for children to provide urine than to do a blood panel. It's affordable, even without insurance. It's between $200 to $500. It's accessible, so people in rural communities do not have to come to town or wherever they have to go to a hospital. You can ship it in from the house with proper preparation, and it reflects the gut and brain axis. So the gut and brain axis is a bidirectional communication system from your brain to your gut. And it plays a significant role in the CSN functioning because it releases neurotransmitters. Organic acid testing can measure bacteria overgrowth, oxalate levels, carbohydrate metabolism, energy production, ATP production, amino acids, byproducts of neurotransmitter metabolism, vitamins, trace elements, antioxidants from food, detox pathways, oxidative stress, and mineral markers and cofactors. This is significant to the clinical setting because OAT can identify different subtypes of stuttering. There are many different subtypes of stuttering, which requires a more personalized treatment, and also shows the association between biomarkers and stuttering, such as thiamine, histamine. And in conclusion, stuttering is a neurological complex. Current assessments focus on symptoms and not the causes. O offers a biological insight, and if we use O as a tool, it can improve the diagnostic accuracy and treatment effectiveness. And these are my sources. And thank you, Dr. Snyder, for your help.
Next up, we have Ms. Kennedi Stancil from Jackson, Mississippi. She's an allied health studies major from University of Mississippi. And her topic is the influence of NFL concussion protocols on parental decisions in youth football participation. So let's give her a hand.
Good morning. My name is Kennedi Stancil. I am a rising senior here at the University of Mississippi. And this summer I conducted my research with Dr. Kristen Austin. And the title of my research, once again, is the influence of NFL concussion protocols on parental decisions in youth flag football participation. So, football is one of the most popular sports in America, but there has been growing concern over player safety, specifically when it comes to brain injuries and concussions. The media, on the other hand, plays a role in these safety concerns. They have been covering high-profile cases such as Junior Seau and Aaron Hernandez, who were both diagnosed with CTE or chronic traumatic encephalopathy after their deaths. And these rising concerns over chronic traumatic encephalopathy and CTE and concussions have been influencing whether or not parents are deciding to enroll their children within tackle football versus flag football. Concussions are a traumatic brain injury that happens after a blow to the head, and one of the leading causes and one of the leading issues or one of the leading diseases of concussions is CTE. CTE is a progressive brain disease that happens after repeated traumas to the head, and these issues are bringing awareness to why or why not parents may decide to enroll their children within flag football over tackle football. To address these concerns, the NFL has adapted NFL concussion protocols. These protocols were adapted in 2013, and some key features of these protocols are sideline evaluations, return to spotters, and return to play rules. But despite these improvements, concussions do still happen. In fact, 182 concussions were recorded this past NFL season during the 2024 season. So we still see concussions on the field, and this is why there is a rise in concern of whether or not parents would like to enroll their children in tackle football. The media, on the other hand, plays a role in how the public views concussions. Some people may feel like it's an over-exaggeration of concussions or CTE. Some people don't even believe in CTE, but there is coverage of CTE with players like Aaron Hernandez, former NFL players like Aaron Hernandez and Junior Seau. On my slide, I included two newspaper clippings of them both. The first one is showing Junior Seau. Junior Seau committed suicide, and he was genuinely a happy person. His family believed that he was not depressed. He was not angry or anything, which are two really big behaviors that are shown within CTE. But on the other hand, we have Aaron Hernandez. The newspaper clipping is entitled "Sick Mind" because Aaron Hernandez did commit a murder, which landed him in prison, where he later then committed suicide. He was diagnosed with CTE after he committed suicide, and it was found that he had one of the most prominent to date cases of CTE at just the age of 27. And because of this, there has been a rise in flag football participation. In fact, since 2020, there has been an increase by 39% of the participation within flag football, because there has been information that shows, of course, Flag football has fewer head impacts, but of course, it's still not risk-free. Head impacts still happen, but parents do see flag football as a safer option. All of this led me to my research question, which is, does media coverage of the NFL concussion protocols influence parents' decisions to enroll their children in flag football instead of tackle football? One of the reasons why this matters is because, for example, the son of Pro Football Hall of Famer Ray Lewis, he had a son, Ray Lewis III, who passed away in 2023 due to a drug overdose. When he passed away, it was found that he had CTE at just the age of 28, and his mother has since developed a foundation where she has been trying to conduct outreach to give families to realize the dangers of enrolling your child in tackle football at a young age. Her son started playing tackle football at just 5 years old, and she feels that if he did not play tackle football so early, he may have avoided getting CTE later. She thinks that he should have started playing at 14 years old instead. So the reason why this matters is because for the evolution of football and for parents to feel comfortable and safe with their children playing, it is important that they know the risk of playing tackle football at a younger age. And in order to conduct my study, I did an IRB approved study where I conducted one interview of a parent who has a 14-year-old son who plays flag football. I asked 5 questions that were related to concussions, their knowledge of concussions, their knowledge of CTE, their knowledge of concussion protocols, and etc. And I quoted that interview for quotes in order to understand better why she decided to enroll her child in flag football. And as a result, the parent was more so willing to enroll her child in flag football, not due to the risk of concussions, but more so due to the risk of her, not risk, but due to more so why there was not a tackle football program in the city of Oxford. Her son is 14 years old, so I'm not sure why she's choosing to not enroll her son in either middle or high school football, depending on if he's in middle or high school. She also feels that the year weighs her son down. He is more on the lankier side, as she says, so she feels like flag football is better for him. She's aware of the NFL's efforts to talk about concussions and their concussion protocols, but she does not believe that they're making a good impact. And of course, she believes that the media does make concussions look bad because she's seen the games before. She watches concussions. She's seen a concussion happen before in real time. So she does believe that the media does cover and make concussions look bad. So what does this all mean? This means that the media does play a big role in shaping the perception of how parents view concussions and the NFL's concussion protocols, and also how they view tackle football. There needs to be more done to inform these families about the risk of playing tackle football. And some limitations of my study, of course, are that there was a small sample size. I was only able to interview one parent, but in the future, I would like to do more interviews and also compare backgrounds of tackle football and flag football to learn more about why these parents decided to enroll their children in flag football versus tackle football. And in conclusion, the NFL safety measures are not enough to ease parents' concerns over tackle football, and the media narratives will drive fears and influence parents' decisions to continue to enroll their children in flag football over tackle football, and it will continue to be on the rise to put them in flag football. Also, I would like to thank my mentor, Dr. Kirsten Austin. I would like to thank all of the people that helped with McNair, and I would like to thank my cohort, of course, and I will open the floor to any questions. Yes, she's
Dr. Stevens.
The mother, I'm grateful with her that if she wished that her playing football at a later age, sort of at around 5 years old.
Yeah, he started playing at
so. Is there anything research that indicates that starting team football at a later age, the lessons about developing CTE or any type of traumatic brain injury.
I'm not sure if there's any research, but I would assume that there is, being that she feels that way, that she could have enrolled her child at 14. I don't want to give you the wrong information, but I can look that up for you if you want me to.
That's another hand. Oh, Bryant.
Like for instance, a tax have a professional school district, well, like the very like they're living in town. That is most
The decision of to enroll. Oh, yes, for sure. Because, for example, like the parent said, there isn't a tackle football team here in Oxford, even though her son is either middle or high school. So there is a tackle football team, but she says that she didn't enroll him because there isn't a football team here. So I definitely do believe that that is a reason.
Austin.
Future goal. My future directions or goals. It informs my future directions or goals because eventually I still want to continue doing research more primarily on CTE. I do want to keep it within children, but I think I want to change the age range. I would probably want to go further down to 5, between 5 and 10 maybe. So it'll really help my research in the future.
And that will remain in the sports field.
Yes, in the sports field.
All right, well, if there are no more questions, give Kennedy another hand. All right, next up we have Miss Karmece Talley from Hopkins Park, Illinois. She's a computer science major at Russ College, and her research topic, very timely, can AI detect its own work, evaluating large language models, self-detection, and computing education. Let's give her a hand.
Hi, everyone. My name is Karmece Talley. I am a senior at Russ College in Holly Springs, Mississippi. It's an echo. Give me just a second. I can use this, right?
Yes.
Okay. So before I start, I want everyone to just take a thought on AI and just analyze not really the history, but just like how it has helped you in your academic career or even just your personal life. So again, my research question is, can AI detect its own work, evaluating large language models, self-detection, and computing education? So through my research, we basically went through and we decided whether like ChatGPT, Claude Sonnet 4, Gemini 2.0 Pro can basically detect its own work. So what we did was we formulated like 25 prompts so that we can basically send them through those large language models and determine if they can be detected if it's AI or human written. And why does this matter? This matters because during as we know that the world kind of heavily resort into using AI instead of using their own mental capacity to just do the work or even just get things done when it comes to resumes or cover letters or even just things in general in your life, like recipes or anything like that. So the main thing that we decided was using these 25 prompts, sending them through the different large language models. We basically just asked them different questions like, can they sorry guys. Just a second. Okay, so yeah, we basically just asked them different things like for like algorithm or descriptions of technical concepts, the documentations of code, and like just different concepts that allow them to answer whether it's AI or human written. So again, we use ChatGPT, Claude, Gemini, and these basically, when we did the process, ChatGPT kind of gave us like not really like a perfect answer, but more so like a answer that we really can't use on different like tests, or just making it easier to where we can't actually determine if it's AI or human generated. During our findings, we kind of like, if as you can see here, this is here, the baselines was basically the human written prompts, and when we put them through like ChatGPT, as you can see, it kind of disappointed us here because it gave us mostly AI, and then here, even though it was mostly human written, it still gave us only 68%, and then during Clyde, it gave us 72%, so that's also high. And then we did two different phases, which was the default here and then the deceptive here. Default gave us mostly human written, and it kind of gave it almost 100%, but not nearly like close to 100%. And then Gemini gave us 100%, and then Claude gave us 100%. And then during the deceptive phase, we got 63%, and then 35%, and then 39% for Gemini. In our interpretations, we basically decided or realized that these AI models cannot always be reliable when it comes to submitting work and kind of giving us like sources that they think of on their own versus giving like actual answers that help with whatever you need, whatever you ask it. And it kind of tricked its own self during this process as well, because it kind of like whenever we asked it was it AI or human written, it gave us like for some of the human written, it gave us like, oh, this is AI, or this is a human written, when like we knew like based off what we had written that it wasn't AI or it wasn't human written. And it doesn't give honest feedback all the time because, like I said, it lies, and whenever it gives the feedback, it's not like the actual feedback. It's not accurate at all. So in conclusion, like I said, it's not reliable. It's not reliable, and it can't detect its own words or the formulate outputs. So the outputs, like I said, they weren't like legit, and it wasn't giving like full, like honest answers. And it kind of fools himself whenever it comes to like making it to where you get the honest answer and you can use it for whatever you were asking them in the first place. Questions, answers.
Is it letting you tell here?
Oh, I don't see nobody out there now.
Okay, let's acknowledge them now.
So I had the pleasure of working with Dr. Christina Trotter and Dr. Christopher Berger. They were a major help in this process because I've never done research, and they made it easier to where I could, get on whatever they had going on before I got here. And yeah.
And Dr. Trotter is on Zoom. I saw her name, so she is watching.
Oh, hi, Dr. Trotter.
Do we have any questions? I'll go first. I was in the conference Monday.
Yeah.
And they were talking about lawsuits across the nation with students suing because they got kicked out of school because it came up as plagiarized, but it really wasn't plagiarized. And so in your research, did you see where anybody's working to maybe build some sort of more reliable AI detection, or is there even a thing that can be done?
Yeah, so it's kind of iffy because like even when like overall it's AI. So it's like you can't really like, there are different like platforms where you can detect it easily because people use them a lot when it comes to like tests and like coursework in general. But for the most part, it's only limited like models that can actually help detect it and find out different ways to kind of prevent it in a way. But it's really iffy. It's not many at all. Yeah.
Any other questions? Yes, Dr. Brown.
Yeah, so when we ask them the different questions, we ask for the confidence level and we ask for the reasoning. Most of the reasoning was like, oh, it's formatted to how humans think, or it's kind of formatted to what ChatGPT would format its wordplay in a sense. So it kind of really gave explanation because we asked it to give explanation, but for the most part, it just gave us like because of the format or because of how the wordplay was.
Any other questions? All right, well, thank you so much, Carlos. Very good job. Next up, we have Ayanna Williams from St. Louis, Missouri. She is a chemistry major at Russ College. She's also Miss Russ College. So her research topic is experiencing the evidence, how forensic chemistry internships shape student perspectives. So let's give Ayanna a hand.
Hi, everybody. My name is Ayanna Williams. I am a senior chemistry major from Russ College. And my mentor, she is not here, but her name is Dr. Abby Boyd. And I want to thank her. Let's see. Nope. Okay. Today, we are going to be talking about experiencing the evidence, how forensic chemistry internships shape student perspectives. So my project overview, I'll be going over my research question, my literature review, my research method, my data and results, and my conclusion. So to start off with my research question, how do forensic chemistry majors describe their experiences and outcomes from a summer internship? Well, to be able to answer this question, we first have to understand what internships are and why internships are so important to student learning. So can anybody tell me what an internship is? Yeah. Or in other words, it's a short-term experience that students do to gain skills and explore careers. So can anybody explain why they are so important to student learning? Y'all can both answer if y'all want to. Yes. Okay, yeah. And I also said they build confidence and career readiness. So perfect. So my research method, the framework I used was the expectancy value theory. The purpose of using this framework was to identify ways to improve internship design and student support. And it also shows how confidence and perceived value impact student engagement and outcomes. So what is the expectancy value theory? Expectancy is the student's belief in their ability to succeed or what's being expected of them. And then the value is the important usefulness or interest placed on a task or what they felt they gained from that experience. So this is what I did. We had 6 forensic chemistry students that wrote 6 reflections from their internship experience. And to get my data or my themes for my project, we did coding, 2 different types of coding. So the first one was emergent coding, and the second one was consensus coding with a second researcher. And when I did my coding, I organized them into two different categories, which one is expectation factors and two value factors. Okay, so I'm gonna try to break it down so y'all can see like the process that I went through while I was doing it. So this one over here is an example of emergent coding. So as I went through the reflections, I used the green one to highlight the expectation factors. So anything that was being asked of them throughout like their process. And then in yellow, no, sorry, I switched it. Yellow is the expectation factors and green was the value factors, what they felt they gained from the experience. And then pink was just like extra notes that I thought were important and I may have to circle back to. And then this one over here is consensus coding. So this is what I did with the other researcher that's also doing a similar project to me. We split them into the two sections, which I explained, the expectation factors and the value factors. And over here, to keep track of them, we wrote the initials of each reflection. And then we just found all the similar themes within them. So I came up with 4 expectation factors. Job shadowing, career exploration, research and data analysis, and workplace preparation and management. So job shadowing is like observing a professional on their day-to-day like job duties and responsibilities. Thing 2, career exploration. So the students weren't told to just stay like in one certain place, like they were able to go in different places within the forensics lab to kind of see like all the different career paths and how they all kind of combined and help each other. My third expectation factor is research and data analysis. So kind of what we're doing now with doing our research papers and stuff like that. Workplace preparation and management. So that could be anything from like learning how to prep a lab, filing paperwork, stuff like that. And next, I'll be talking about my value factors, and I found 5 of those. So we have real-world applications of chemistry, networking, practical lab experience, professional growth and development, and planning ahead. So real-world applications of chemistry, that is basically students described how they took what they learned in class and applied it to what they were doing in the actual lab. Networking, that's actually another thing that we're practicing right now. Meeting peers, mentors, professors, actual, professionals that do the job. Practical lab experience, that is any like hands-on experience. So whatever they did like in the lab as far as their experiments that their mentors did or took them through. Professional growth and development, that was anything dealing with what students felt like they gained. So professional skills, talking skill, all of that. And then planning ahead, a lot of the students stated their future goals and said how they wanted to pursue like going into grad school or continuing, their education, or actually finding what career path within the forensic chemistry lab they wanted to go down. So, in conclusion, internships are beneficial for students Two, this framework revealed how confident they were and what they gained from it, which was the belief and value, expectation and value. And lastly, most importantly, we can use these insights to redesign the curriculum to include more real-world skills, preferably designed by major or like interest. Bridge the gap between classroom learning and professional expectations. So basically connecting better with, different organizations and institutions that offer these, internships to students. And lastly, help the school to career, like, what am I trying to say? Yes. Making a easier transition into, yes, the workforce. And that was all. Thank you.
All right, any questions? Oh, acknowledge your mentor.
Oh, she's still not here, but
What's the name?
I just want to thank Dr. Abby Boyd and everybody here, my cohort, Dr. Howard, Dr. Harrington.
Thank you, teacher. All right, Ms. Vincent has a question.
Can you rephrase the question?
So she was saying, like, with your internship experience here, how did that relate to your project? So like you were looking at how internships in that field impacted them. How did yours impact you, basically?
Mine impacted me a whole like different way, I guess you could say, because this is like my first time actually doing like a deep dive into a research like project in like a formal research paper. So it's taught me how to write a research paper, how to a lot. I could go on for days, but it's taught me a lot of professional skills for sure.
Dr. Cochran.
Oh.
So when you look at the 6 students who were part of internships, were you able to that their assessments have were you able to actually work with students? Because some have allowed so what's your experience as far as review tracking?
I'm actually glad you asked that because I did forget to mention. These reflections were written around 2021, around the time COVID happened. So I did note that one student, he felt like, or he or she, I don't know, they were anonymous, but they felt like they didn't get the opportunity to do hands-on work and they wish that they got that experience more, similar to me, compared to other students that actually got to do hands-on stuff and got more opportunities. So I feel like COVID could have been a factor. And yes, like I know how some labs are probably more stricter. So.
Yes, that one.
Hi. You had highlighted the expectation factor and the value factors that are most prevalent or most relevant. Now, listed for and listed by what was like the high expectation or the top value.
I feel like to me, I feel like the biggest one for expectation factors would have probably been workplace preparation and management. And it could kind of probably tie with job shadowing as well. Because a lot of people, like a lot of the students mentioned both of those a lot. So, oh, and then the value factors. I mean, I feel like all of those are important because those were the things that they felt like they gained the most from the experience. So I feel like all of these are important to students, especially me, because I know all of us are actually experiencing these things currently. So yeah.
Any other questions? All right, well, let's give Ayanna another hand. Move to our final presenter, last but not least, Kendall Zollicoffer. Kendall is from Madison, Mississippi, and also one of my former students when I was a school counselor. She's from Allied Health, Pre-Nursing, University of Mississippi, and her topic is emotion regulation among African-American adolescents experiencing grief, a narrative review. So let's give Kendall a hand.
Good afternoon, everyone. How are you guys doing today? That's good. My name is Kendall Zollicoffer, and I attend the University of Mississippi, and my mentor was Dr. Laura Dixon. She's not present today, but she's on the Zoom. My topic, once again, was emotion regulation among African American adolescents experiencing grief. The inspiration for my work was my grandparents. I lost my grandfather at the age of 14, and shortly after, 3 years later, I lost my grandmother. And they're very influential in my life, and it was very difficult grieving at this young age, but I was blessed with a great support system. And yeah, so it's my grandfather, these are my grandparents, and then this is my last photo with my grandmother before she passed. So, I have a question for you guys. If you feel comfortable, raise your hand if you've ever experienced grief. And I would like you guys to think about how you felt during that time. My research question and objective is, what does emotion regulation look like among African American adolescents who experience grief? My research objective is to identify directions for future research, including additional studies on how emotion regulation works in African American adolescents and the potential need for culturally informed interventions such as medication or professional help, such as therapy. My overview would be normal grief versus complicated grief, grief in adolescents and African Americans, emotional regulation and grief, emotional regulation, African Americans and adolescents, and the ending is conclusions and future directions. Okay, losing loved ones. Study has shown that at least 96% have lost a loved one. Grief is often universal, but it can be shaped culturally and like your identity. So we all know that death is inevitable. You're gonna die at some point. But I'm studying adolescence, and it is crucial. This research is crucial and important because many adolescents are often overlooked, and most aren't able to properly regulate their emotions. And how can they if no one ever talks to them about it, or they don't have a trusted adult to talk it through with them? Bereavement and grief. Oftentimes, many people think that this is the same thing, but I'm here to tell you that it is not. Bereavement is the experience of losing said loved one, whereas grief is the emotional response after you have lost said loved one. An example, it means that, okay, bereavement, you have lost your loved one. You understand that your loved one is gone and at peace and resting. But grief is the emotional response after you have lost that loved one following hurt, anger, or sadness. Okay, this is another thing that many people think are the same. Normal grief versus complicated grief. Normal grief can last up to 6 months and is typically more mild. And symptoms can include you're able to understand that your your loved one is gone and you're able to cope with it and have healthier coping mechanisms. Whereas complicated grief is more persistent and more severe, and studies have shown that at least 9.8% of adults will experience prolonged or complicated grief. And symptoms can include longing for your long loved one, yearning, and possibly just not being able to get over the fact that your loved one is gone. So this can lead to unhealthier coping mechanisms and possibly mental health conditions. Okay, grief in African American adolescents. This is important because many are understudied and underrepresented, and there's not many research that has been done on this particular topic. But what I have found is African American adolescents face higher exposure to loss. And also, while they are experiencing that and they're not able to talk to someone and have professional help, this can lead to disrupt emotions as far as not talking about it, suppressing your emotions, and so forth. And there also can be some social and cultural factors included, such as you only trust your pastor or you don't trust the healthcare system. There's a mistrust I found in research. There's a mistrust in the healthcare system. So whether most people do not go to healthcare professionals and they feel like they just rather will talk to their peers and get help that way. And then lastly, African Americans have also reported higher levels of complicated grief symptoms. Okay, I'm guessing you guys are wondering, what is emotional regulation? Emotional regulation is an individual having the ability to modify their emotions and formulate coping mechanisms to learn how to manage one's emotions. This can vary across many stages of life, from infancy to adulthood, and if it's not talked about or peers seem like they are not getting enough help, this can lead to poor emotional regulation, and this can also lead to mental health problems such as PTSD, depression, and even anxiety. Unlike other factors such as age and race, emotional regulation can be modifiable. Okay, emotional regulation and grief. Emotional dysregulation has been associated with bereavement and grief. It ties together in a way as if there's psychological symptoms in both. There's lower, and higher. The lower ones include mindfulness, which is being one with your mind, understanding what you're going through and talking to someone. Problem solving, you're able to talk through it, get any additional help that you need, and solve the problems that you have, such as grief. Then cognitive reappraisal, being able to acknowledge what you're going through in a healthier way and everything. Then you have the maladaptive strategies. This is higher psychological symptoms. This can include emotional suppression, so suppressing your feelings about your loved one being gone, not talking about it, or avoidance. This goes hand in hand, and rumination. Yeah. Emotional regulation in African American adolescents who experience grief. Research has reported that African American adolescents have experienced grief and higher emotional suppression rates associated with maladaptive responses. And this is things such as emotional regulation and emotional avoidance. Research has also shown that African Americans, adolescents, talking about emotional regulation and being informed about it plays a direct role in the grief response. Another study has shown that emotion dysregulation, and in particular avoidance, rumination, suppression, had larger effects for predicting mental health problems and outcomes of children and adolescents. Also, again, has been known to be linked to numerous psychiatric disorders. Okay, following again with this slide, there has been reported high rates of PTSD and worse PTSD symptoms in African Americans, and there also has been a high rate of co-occurring PTSD and depression in low-income Black women. Lastly, there has also been reported high rates of psychological symptoms in Black participants, studies have shown. My conclusion is African American adolescents may be at a higher risk for developing complicated grief. There has been extensive research, but like I said, it has been limited on the studies of emotional regulation and grief within African American adolescents. And then also, it would be best if future researchers can find the need to understand how to best support African Americans who are grieving, adolescents who are grieving, and need any support. Also, researchers should investigate the cultural factors and coping strategies. And my future research directions, I would like to examine modifiable predictors of complicated grief, such as finding helpful ways to help African Americans cope with their grief. And then lastly, culturally responsive interventions, such as finding the best treatment that can target and help African Americans cope, such as medicine, art, and professional help. And also future research is needed to develop and test emotion regulation based on interventions to help cope with grief. Thank you so much. And I would like to thank Dr. Laura Dixon. She's on the Zoom, and she has been very awesome and incredible. I enjoyed doing research with her, and also her graduate assistant, Cheyenne and Blaine. Thank you guys so much. Oh, is there any questions?
Any questions for Kendall?
I had 3 kids. They were 4 and 8, and their dad died, and then my husband died, right?
Yes, ma'am.
Being back, that's in 2003. What, and we experienced grief, but what can you have African Americans, what do you think we can do to have African Americans realize that psychological, they be psychological impacted, and how can we get them to realize they probably need therapy at that young age and not when they get older?
That's a good question.
So yes, that's a very good question. So far the research that I've shown is it is important to introduce your child to grief. Like if they haven't grieved over someone, it's good to introduce them to grief. Like talk about it, help them express their emotions. But kids don't understand. So the research that I've done is parents talking to them about it, asking them questions, but also being a listening ear to them has helped them in the future for being able to express those future emotions to their parents, peers, a therapist, but also introducing therapy at a younger age too. Personal disclosure, I have nannied for some parents, and they have started putting their children in therapy at a younger age, and I have seen significant results in doing that, but also them talking it through them and having them feel comfortable. So it's best, research has shown that it is best to start at a younger age to talk about grief, therapy, or any other things that goes on with grief with your children earlier. And if it happens in adulthood or young adulthood, it is best to reintroduce them to it, find ways to help them in the way they would want to help, because at that point they have their own mind or they're grown. So it will be best to give them those resources, and it's up to them if they want to do them. You can't make them. So yes. I hope that answered your question. This. Yeah.
Like as a percentage to like that that impact is you think that looks different in different children, like the stereotypes of like specifically like not express emotions or how person there navigate that.
Right, so that is a great question, but there has really been limited research on the male factors. So far I've seen studies on only females and then of course other races. I haven't really seen any with Black African American males. So that would be something I could possibly do research on in the future. So yes, I love that question. Is there any more questions?
I just wanted to, that was gonna be my comment as well. I would love to see this as an intersectionality study along the lines of race and gender.
Yes, ma'am. Thank you.
Well, there are no other questions. Let's give Kendall a hand.